For a compound to appear colored, it must absorb light in the visible region of the spectrum. So as you probably already figured out from working with organic compounds in lab, most of them are colorless. White solids or colorless liquids because they don't have any absorption in the visible region. But if there is enough extended conjugation, so we've seen in the last video how as you increase the number of conjugated double bonds, the wavelength of absorbance moves to longer wavelength, right? If there's enough conjugation, eventually that absorbance will move into the visible region, which is of course the region from 400 to 700 nanometers. So a good example of this would be the compound Beta-Carotene, which is responsible for the orange color of carrots. So there are 11 conjugated double bonds in beta-carotene, and the lambda max for this compound is 497 nanometers. Now, why is this compound orange is the next question we need to consider. Well, in order for a substance, any substance for that matter, to appear colored to us, substances will appear colored because the wavelengths of light that are not absorbed are reflected off of that substance and reach our eyes. The wavelengths that are absorbed by the compound. They are not reflected by the compound and they do not reach our eyes. So what this means is if we are looking at beta-carotene, we are not seeing any of the light at 497 nanometers or even 30 nanometers on either side of that. So what we need to do now is look at a color wheel. Remember a color wheel from elementary school art class maybe? So this particular color wheel actually correlates colors with wavelengths. So as you could probably imagine, the ultraviolet comes next to the violet in the grand scheme of the electromagnetic spectrum. So the violet is the region from about 400 to 430. Then the blue region is 430 to 490, etc. So those are wavelengths that are being absorbed. So if something is absorbing light at 497 nanometers, it's absorbing blue-green light. So then what reaches our eyes is everything except blue and green, which for all intents and purposes is going to appear to us to be the complementary color to what is being absorbed. And the complementary color of blue is orange. That's the color that's directly across from it in the color wheel. Therefore, compounds that are absorbing in the blue are going to appear orange. Compounds that absorb in the green are going to appear red. Compounds that absorb in the violet are going to appear yellow. So sometimes you do see organic compounds that have some yellow color to them, and that's because they're either the compound itself or some impurity in the compound does have an absorbance that just makes it into the visible region. So notice that every color change corresponds to a change of about 50 nanometers in wavelength. So now I want to talk about a couple of examples of colors, and one of them that's actually pretty important are acid-base indicators. So you've all probably at some point in a general chemistry course done a titration of an acid with a base where you took a little bit of this indicator, usually phenolphthalein, put it in there, and when you've added enough base, you're looking for a pink endpoint. But you probably have not considered at all what this substance called phenolphthalein is, nor were you in any position to consider that when you were taking general chemistry. But compounds that can serve as acid-base indicators need to be compounds which undergo a reversible structural change depending on pH, and that the form present at higher pH and the form present at lower pH have different colors, which based on what we have presented in these videos means that they have different amounts of conjugation. Okay, so let's look at phenolphthalein. Phenolphthalein is colorless at acidic or neutral pH, basically below pH 8, and it's pink in basic solution. So if you look at the colorless form on the left, there are 3 aromatic rings, so it will absorb in the ultraviolet, but it will not absorb in the visible, therefore it will appear colorless. And what you need to look at is that the central carbon is sp3 hybridized, okay, the one bonded to the 3 aromatic rings and the oxygen. And therefore there is no extended conjugation here. So what happens when you take this molecule and you put it into base? Well, as we have seen already in some of our mechanisms, under basic conditions, things which are not a very good leaving group under neutral or acidic conditions can become a good leaving group under basic conditions. And the carboxylate anion is a sufficiently good leaving group that it leaves the central atom. And then the effect of that is to actually form extended conjugation. So that central atom ends up forming a double bond. You know, what would be a carbocation that's left on that central atom ends up forming a double bond. You convert an OH into a C double bond O, and you end up having extended conjugation over 11 double bonds. Therefore, the compound appears to be colored. At higher pH, you know, the carboxylate anion will come back in, reform the structure that is present. Sorry, at lower pH, reform the colorless structure. Another application of colors are molecules that are used as dyes. So long time ago in history, very few people wore colored clothing because dyes had to be extracted from natural sources, and they were colored clothing was really expensive. The advent of synthetic dyes in the late part of the 19th and early part of the 20th century was one of the major breakthroughs of organic chemistry. And what I have drawn here are a number of dyes: methylene blue, methyl orange, aniline yellow, malachite green, alizarin, and approximately they are drawn in what color these dyes actually are. You can actually synthesize methyl orange in the laboratory. They do that at some schools, and then you use the methyl orange that you synthesized to actually dye fabric. One more application of color, though, that's actually more biologically relevant are fluorescent compounds. So most colored compounds are colored because, as we have seen, they absorb light in the visible region of the spectrum. But some compounds actually emit visible light, and these compounds are fluorescent. So why do certain compounds fluoresce? Well, as we have discussed, after a molecule absorbs light, an electron gets promoted from the ground state to the excited state. So the excited state that the molecule finds itself in is higher in energy than the ground state. It has absorbed some energy. And the molecule has to do something with that energy. It can't just sit there in the excited state forever. So most molecules release that energy. Well, they all have to release that energy to return to the ground state, and most molecules release that energy in the form of heat. However, a few molecules that have certain specific chemical structures will actually release that energy in the form of light by emitting a photon. So an example that I am personally familiar with is a molecule called alexafluor 488. So molecules that fluoresce are called fluorophores. And this structure can be bound to many biologically relevant molecules such as DNA and proteins. It's actually the top part of this structure that's responsible for the fluorescence. The ester on the bottom is how you link it in to a DNA molecule or a protein molecule. And this molecule absorbs light at 488 nanometers, which means it's absorbing blue light, but then it emits light at 515 nanometers, so it is emitting green light. It actually glows green. Now, all fluorescent molecules are going to emit light at a longer wavelength than they absorb light at. That's due to the fact that they've released some of their energy through vibrations before they have a chance to emit the photon, so that the photon they emit is at slightly lower energies. Okay, so fluorescent tags are incredibly useful in terms of being able to visualize biological molecules. And there's actually a protein that has a fluorescent tag on it that's called the green fluorescent protein. It is a naturally occurring fluorescent molecule, and it has become incredibly useful in carrying out biochemical assays. Okay, so that's an introduction to how color fits into the world of organic chemistry.